Okay, hello everyone, and thank you for joining us today. My name is Amy, I'm with EAS Consulting Group, and you are joining us for a webinar on understanding GRAS submissions and avoiding data pitfalls. Today's presenters are Bob Kapp and Robin Guy, and we are very pleased to have two esteemed toxicologists and EAS consultants with us. Just a quick note for everyone out there, if you have any questions during the webinar, you're welcome to write them into our questions box. And I will start the presentation by turning it over to Catherine Sacra. She is EAS Director of Labeling and Cosmetic Services. And she'll introduce our two speakers. So Catherine?
Good morning, everyone. As Amy said, my name is Catherine Sacra. I manage our labeling and cosmetic consulting teams here at EAS, and that also includes our fantastic food additive team. I want to welcome you to our webinar on GRAS submissions and avoiding data pitfalls. And first, I'd like to just say a couple of words about EAS. For those of you who are new to EAS or have only worked with us in certain areas. EAS is a premier consulting group, and we specialize in providing FDA regulatory consulting services to the food, dietary supplement, cosmetic, pharmaceutical, medical device, and tobacco industries. We've recently added cannabis and hemp to that, given the passing of the Farm Bill. Our extensive network of consultants includes both former FDA officials and industry experts. As this webinar concerns some of the issues surrounding GRAS notices and independent GRAS conclusions, I would like to highlight our background in this area. If we could switch to the next slide, thank you. Since the inception of the GRAS notification program in 1998, EAS and its predecessor, AAC Kendall, have submitted over 20 GRAS notices to FDA, and we've prepared numerous independent GRAS determinations for our clients. In addition to that, we provide regulatory consulting for other types of FDA submissions, such as food contact substance notifications, health claim petitions, new dietary ingredient notifications, etc. So I just, if you have any questions on any of those services, feel free to reach out to me at any time. I'm pleased to introduce our speakers for today. Our first speaker, Dr. Robert Kapp, has over 30 years' experience as a toxicologist involved with the management, development, and safety of new and existing products in a broad spectrum of industries, including preclinical study design, study reports, occupational and industrial toxicology, and the evaluation of clinical and program safety data. Prior to becoming a consultant, Dr. Kapp worked as the director of the Toxicology Laboratory at Exxon Biomedical Sciences. He holds a Ph.D. in toxicology from George Washington University. Our second speaker, Robin Guy, is a board-certified toxicologist and quality assurance consultant with more than 30 years of experience in both industry and working as a private independent consultant. She has worked in the pharmaceutical toxicology and food ingredient safety assessment area. She's an expert in clinical toxicology, regulatory and good laboratory practices, designing and coordinating toxicology studies and programs, as well as preparing regulatory submissions. Ms. Guy worked with the NutraSweet Company, Monsanto, and with J.D. Searle and Company.
Good afternoon. I just wanted to give you some background on this. It's usually a challenge to try to come up with, to discuss this topic for such a diverse group. So to get on the same page, basically, what I wanted to do is explain a little bit about government and how it operates in legislature. I think we all know this, but the government generally operates by a crisis. Civil War, World War I, Depression gave us a New Deal, World War II, the financial crisis of 2008, we got bank bailouts. Now we have immigration and gun control, not quite fixed up yet, but there's nothing different with that and how the FDA has to work. It responds to public outcries and what have you. So along comes a guy in the early 1900s by the name of Upton Sinclair, and he's writing a book. He's trying to explain how food processing was filthy and how it's dangerous labor practices and cruelty to the animals in the stockyard, but really he was pushing socialism. So he went undercover and was writing graphic descriptions of the circumstances and came out with this book. Now, when I say graphic, I mean graphic. Men were falling into the vats. A lot of times they weren't recovered. The food was still processed. It was like the Middle Ages. It was completely unsanitary and horrific situation. So he publishes the book sometime in the summer of 1906. President Teddy Roosevelt is reading the book. Now, he's a big breakfast guy. So sausage and eggs, and he read the book. And the story goes that he threw his sausage out the window because he was so disgusted with the situation there. So the book and Teddy Roosevelt were the primary impetus for the passage of this Pure Food and Drug Act. The next real crisis came in 1937. 1937, elixir sulfanilamide was a new wonder drug. It had been developed by a company called Messengill, and the problem with it was it tasted bad. They couldn't get it to dissolve in sugar solutions or honey or what have you, so their chief chemist, a guy by the name of William Hawkins, he decided to add a little diethylene glycol because he knew it was sweet. It's also known as antifreeze. Now, that sounds horrific today because we know better. Back then, they didn't. So they made 240 gallons of this drug and they shipped it nationwide. And almost immediately, people started dying. 107, mostly children of all things, died. It was recalled. The FDA jumped in, they snapped into attention and got 234 gallons in 1 pint of this stuff back. So all of this damage was caused by less than 6 gallons of this product. In typical fashion, I guess the company disavowed, this would be Messengill, they disavowed any responsibility because the toxicity of antifreeze was not widely known and no safety testing was done, or get this, required by the FDA. So while the company was disavowing any responsibility, Watkins committed suicide, so he must have felt pretty responsible. As a result of this crisis, that Food and Drug Cosmetic Act, 1938, was passed. In 58, that was the next crisis. Now we're going from one crisis to the next. The 1938 law really wasn't sufficient, and it did not address food additives. Color additives were being added, and some of them were actually poisonous. Those of us who lived during that time, at any rate, it was a big thing. It was all over the news. Some of them were even carcinogenic. They also developed a definition for food additives and GRAS. So all this was passed, and the crisis is perceived by the colorants and food additives in products. The next was the thalidomide incident. While it's not really directly related to GRAS, it's an interesting situation. It was actually handled well. The crisis didn't cause a crisis with the public. The only crisis was with Richardson Merrill, who was trying to get this thing passed. They sent 6 requests to the FDA without data requesting that it be approved. And Dr. Frances Kelsey, rest her soul, was the only person in the whole line who refused it, and she became a hero after the fact. So what happened was that most doctors in the United States really didn't approve the drug, but some physicians had it, and only 17 children were born with the phocomelia defects, which were common with the thalidomide. That's where they had grossly disfigured arms and hands and legs, hands and feet on the end of very short appendages. Very difficult situation. But on the other hand, 100,000 plus were found in Europe because of the lax rules. Now this resulted also in the Kefauver-Harris drug amendments, which clarified how drug makers were to prove their drug works. Of course, it's a little off the beaten path, but I just wanted to show you that basically what we have here is a situation where crises are making things happen, and they happen sometimes for the good, sometimes for the bad. The consequences of this are that the FDA now regulates 75% of all the things that we eat, and the Department of Agriculture regulates the rest of it. The items that the FDA regulates include the following. All of those items, that is a massive amount of material. That's a $2 trillion market. So that's a lot of work. And you say, well, how much does this cost? Well, it seems like the budget in 2018 was only 5.4 billion, and half of that was paid by the users. So a 5.4 billion budget of the FDA to monitor and control a $2 trillion industry is actually a pretty good bang for your buck, in my view. So going back a bit to 1938, they passed this, which you recall, let me see here, what did I do? There we go. They passed this to replace the Drug Act of 1906 because they found that it was actually insufficient. What they wanted to do is establish standards for foods and drugs and medical devices and what have you that were sold in the US. But the thing that made it different was it provided for federal oversight and enforcement. In other words, it gave some teeth to this, to the FDA, and they could fine you or shut you down or do a lot of things that were not friendly, so people had to get in line. We're going to only talk about the foods part of this. So the foods regulations included standards of quality and fill containers for processed food. What this means is basically, if you had a can and you had it full of flour, you couldn't go and get the dregs of your flour and then only fill it halfway up. If you did that, and believe me, this was commonplace in the 20s and 30s, you could be legally liable for that. You couldn't do that anymore. It set safe tolerances for toxic substances that are already in food. The process, sometimes the processing of food does make things more toxic, and it provided a mechanism for the FDA to set tolerances for those toxicities. Here's the big one. The big one was that you had to prove this stuff was safe rather than have the FDA prove it was adulterated after the fact. So this all had to come beforehand. This was a big change. Another thing that really went over like a lead balloon was the unannounced inspections of facilities. It's commonplace now, but at the outset, this was a difficult issue for most of the manufacturers in this country to get a hold of. No false statements about the food items, you know, the snake oil salesman and all those things. That was all pretty much true. It mandated also another thing where you had to actually put your product name, the quantity, the contents, who you were, or how you could get a hold of you, no misleading containers. So jumping out to 58, this was passed in response to the food color crisis. So many of the substances in food really had to be there. This is basically what it did. There are so many things that are in food. They started by exempting two areas. And the first area that they exempted was the things that had already been determined to be safe by the federal government. That would be the prior sanctioned substances, those things that were looked at by the U.S. government, determined to be GRAS before 1958, which is the date of the law. That's why we're talking about that. Those things are being grandfathered. All right, now there's a whole group of things that they didn't know about, and they figured, okay, well, there's things that could have been grandfathered that weren't because either they weren't there or they didn't have time to do it. So let's give a mechanism for these materials to be exempted if they're really safe. And then they gave a whole series of ways that they could be exempted. Now, the Delaney Clause. Let's spend just a minute on Delaney Clause. It's not really applicable to GRAS, but it's an interesting regulatory issue. It underscores the complexity of the regulations. A guy by the name of James Delaney, and I won't belabor this, but he was a congressman from New York. He decided to say, okay, no chemical that could cause cancer could be in food. Now, at first blush, that seems very reasonable. Who wants to eat something that causes cancer? So in 1958, when they were looking at all these things, there were about 200 known carcinogens. There wasn't really indication that there was going to be a whole lot more. They thought they had their handle on it. The sensitivity of the analytical equipment was very, very limited. So while zero tolerance is a catchy phrase, and we use it all the time nowadays, it is decidedly impractical. First, many carcinogens have been identified since then, so the list has now gone to way beyond 200. The sensitivity of the equipment has gone from parts per thousand to parts per trillion. So if there's a molecule of some carcinogen in your food, it's going to show up, and it has to, by the Delaney Clause, it has to be exempted. So The first, here's an example of how the Delaney Clause didn't work. The first use of the Delaney Clause was in 1959. Secretary of HEW, I think it was Art Fleming at the time, sent out a warning that there was a carcinogen in some cranberries. It's called aminotriazole. Okay, so when did this warning come out? A week before Thanksgiving in 1959. Those of us who were on Medicare can remember that that was really quite a Thanksgiving. Even Mamie Eisenhower got into it. She added to the scare by changing from cranberries to applesauce. So what happened? The cranberry market collapsed. Now, the odd thing about this is, because the Delaney Amendment is all or none, the Secretary failed to explain in the warning that you'd have to eat 15,000 pounds of cranberries every day for maybe 3 or 4 years to have the possibility of any cancer. So this resulted in not only a devastation to the cranberry market, but it was going to start applying to other things, and the FDA saw the problem. So they started changing this from the mere presence of a substance that causes cancer to a significant risk. This was the development of the de minimis rule, which was adapted eventually. The acceptable risk for cancer is 1 in 1 million. Now that may not be acceptable to you, but it's acceptable to the FDA, at least at this point in time. Enough with Delaney. They also, in this 1958 amendment change, they defined the term food additive. Now I'm not going to read this. This is a lawyer's interpretation of what a food additive is. Pretty complicated. I'd say it was stuff in food, sort of like things like sodium and potassium nitrates, the sorts of things that are preservatives that are used, and they've been used for years in food. Another term they actually defined was GRAS, which is generally recognized as safe, the chemical that could be added to the food, determined to be safe by experts and published data in historical use, and that could also be exempted. Examples would be salt, sugar, spice, things, exact same things that you would think would be safe. Now, how do we get to safe food now? Well, there's a couple of pathways. One is the pre-marketing approval process. You can take all of your data, pile it up in a big box and send it to the FDA and ask them to determine whether or not this stuff is safe. And that could take months or years. It's not a preferred way. Another thing is, if it were grandfathered, as I've said previously, prior to 1998, or 1958 rather, there was a whole vat of data that was available to everyone, and that those things would be considered grandfathered, and they would be safe without any further ado. The other thing was, of course, the GRAS, which we've already talked about. This is where there's got to be a mechanism to get some of these things to market. There's procedure what has to be followed. So, GRAS affirmation, a little background. There was another crisis. Oops, let's see if we can go here. That was the cyclamate crisis. This was back in 1958. The FDA had determined that cyclamates were actually GRAS. Then new data started becoming published, and it was showed that rat bladder cancers were increasing, and there were some other things going on, and so they began reevaluating previous GRAS substances. Oddly, because of this data, in 1969, the FDA reversed its decision about cyclamates and took it off the GRAS list. It could only be in dietetic foods. And in 1970, they removed it from approval completely. Can't be in food. Now, to make matters worse, President Nixon decided he was getting into the melee, and they found 400 substances, which he was getting political pressure to find out whether or not these things could be GRAS. So now you have to understand they're getting documents from the public. They're saying, okay, here's our data. We want you to tell us if it's safe. And now we have all these GRAS things that have to be reevaluated by the president and by what we've already determined to be a problem. So what did we do? First thing that they, okay, let's see. There we go. 3 years later, the FDA decided they couldn't handle it, so they hired some consultants to handle it. Fine enough. So from 1972 until 1997, they're getting these affirmations in, and they're looking at all these documents that are being done by this committee, and they conclude they can no longer do this. This affirmation process was too much of a burden for the FDA, very understandably. So they proposed a new process. The process was GRAS notification. It was primarily just to relieve them from having to make all these determinations themselves. How did they do it? They put the onus back on the requester. Let's make the requester make the GRAS determination rather than the FDA. Then the FDA will look at it and decide what they're going to do. Did not require pre-market approval. It's always limited to intended use. In other words, if you make it something, say it's GRAS for this product, it can't be in another product unless you put it in your GRAS document. Recognition, of course, of the procedures and of either scientific procedures or the literature or an expert GRAS panel. Those things are what need to be done to make sure that things are GRAS. All the things that were approved before 1958 were okay to go in. Scientific data had to be in the literature somehow, so there could be a consensus among qualified experts. The common knowledge element, that's what that's called. So, in 1998, they received their first GRAS notification on this interim procedure. And from 1998 to 2016, it was an interim procedure. And the final rule was set in October of that year. What it did is it formalized the procedure of how to put these documents, it formalized the interim procedure of sending the data with your evaluation of it being GRAS. But they also noticed that while all the documents they were receiving were okay and they were fine, they were dependent on the writer and where things were. And it was very difficult for them to start to look through and find data in different places because of the way it was organized differently from one to one. So they set up a 7-part framework for submission to help them get through all these documents. Right now, as of actually June 14th, 847 submissions have been made. Now, I know for a fact that there are more because I've sent in a couple, 2 or 3 since that date, but they're a little behind on their public list. We'll give them, they've got a lot to do there. Quickly, just to go over the 7 parts, and not really get into too much of the detail. First, you have to sign your life away and say anything you say in here is true. Otherwise, they're going to fine you or take your first bullet or something. Everything that you know about the chemical substance has to be put in part 2. So they want to know all the technical details. The dietary exposure. Dietary exposure is always an interesting situation. It's where you not only have to know the dietary exposure of the product you're submitting, how much of it, of your GRAS substance is in there and how it will be used. You also have to look at the market and see, okay, where else are they getting exposed to this? So all of this has to be taken into consideration. So dietary exposure is a very, very critical piece of this document. It helps us determine how toxic things can be. This part here is basically if it's self-limiting because it's unpalatable or it's very heavy or there's so much of it that it's technologically impractical to get things to be exposed. Of course, anything that was proved before 1958 is already there. The most important part of this 7-part thing was a narrative in part 6 that actually ties everything together. Takes all part 2 and all part 3 and 4 and 5 and puts all this together, and it adds an expert panel review. So it's very important that this is done in such a way so that you're explaining how safe your material is. Part 7 is basically a list where the data are. Some are proprietary, some might not be, some other way. So here's the notification procedure as it stands today. You use that 7-part guidance, which I just went through, which is pretty simple. You retain the data so that they can look at it. Okay, now, one of the things I didn't talk about is basically there's data gaps. Sometimes when you go over this, and Robert is going to address this in a moment, you go over this, you realize that you don't have all the data you need to make the call, or your expert committee says, no, you're going to have to look at this. So you run some studies and you get the studies data, you get the reports. You can't put them in your drawer and say, yeah, they're safe. They have to be, remember the common element? They have to be out so that not only the expert panel knows, the rest of the scientific world knows. You can do this in a couple of different ways. You can put it in a textbook. You can go to these authoritative bodies. Not a good way to go. It's much too cumbersome and it takes forever. The best way to do it is to take the data that you've created, put it in a peer-reviewed scientific journal so that others can look at your data and agree. Generally, if that's a big plus for the FDA, if your data are also agreed that it's safe by other scientists other than yourselves. Where am I? Okay. Okay. There are exceptions, and the exceptions are handled on a case-by-case basis. It's not that much of a problem. And I want to go to the next slide. Okay. FDA responsibilities. Okay, how do they respond? There are three ways. It's very simple. They can say no questions. Basically, you got it. They can also say you're disapproved. Basically, that's that. That's the third category is you can say as a notifier, we want you to cease. If we found something in the data we don't like, we're going to use a different product. We're not sure. So they can stop it. So there's only three things that can happen after you provide the GRAS notification. Okay, next is the level of concern. And one of the things that we're looking at is how do we determine what studies must be conducted? Like I said, Robin's going to talk to you about this in a minute. How toxic are the substances? This is basic toxicology. What's the exposure going to be, and what's the potential toxicity of the material? So exposure and toxicity, those are the two things that have to be looked at. The levels of concern, generally, okay, this is very low tech. Category A, low toxicology potential. The next category would be B, intermediate, and of course, C would be the high toxicological potential. So to put a finer edge on that, what are those? Let's talk about that quantitatively. Quantitatively, we would want to look at the next slide, and that would be the category A would be less than or equal to 50 parts per billion in the product. Category B would be 50 to 1,000 parts per billion. In category C, that would be the highest toxicological score. There's more than 1,000 parts per billion. All of those categories are, that's all the parts per billion, as you know. And with that, I will step aside and let Robin tell you about how to determine what studies you have to do. Thank you very much.
Thank you, Bob. Hello, everybody. So I'm going to talk about non-clinical studies that are needed for food additives and GRAS submissions. And I'll talk about types of non-clinical studies. Well, you could have an understanding as to what those studies are all about. So we'll briefly go into those. Okay. Sorry, I'm trying to advance it, and it's not. Thank you. Okay, so don't try this at home. Don't try this at home or at work or without discussing this with a non-clinical or discussing the non-clinical program with a food toxicologist. You really want to get the input from a toxicologist who does have a lot of experience in the food industry. Even CROs, which are contract research organizations where you may be doing your studies, a lot of those, you can't expect to find toxicology experts who really know the food area really well. There are really very few food toxicologists out there. And most toxicologists may be great with pharmaceutical toxicology programs and studies, and yeah, anyone can read the Red Book, but they won't have the experience of working with the FDA on food applications. So planning studies is really it's an art and a science, really, and you need to have a lot of experience working with that. And I just want you to know that the studies listed are not intended to be considered the final non-clinical program for your product. Every product is different. And so that's why you really do need a toxicologist to come in and work with you. And EAS does have expert toxic food experience. So, we could always help you with that. A lot of these studies are going to be conducted according to GLPs, good laboratory practices, and those are a law. That's in, you'll find those in 21 CFR Part 58, the Code of Federal Regulations. GLPs do assure quality and integrity of the data. It's really important in those studies. And they also allow for accurate reconstruction of a study. There's so much that goes into a toxicology study, and you want to make sure that everything that you do in a study is documented so that somebody can come back and reconstruct the study. And it's not just the study itself. It's things that go into the study, like personnel and training and equipment and validating equipment and that type of thing. So you really want to make sure that happens. And also another thing is to assist with the approval of products that are safe. That happens too. So as Bob was talking about the categories, and I just want to go through these fairly quickly. I'll get into more detail on these studies coming up, but right now we'll just talk about briefly what needs to be done in each of these categories. Category A is the low toxicological concern, and you just need genetic toxicology testing and short-term toxicology tests. And then for category B, genetic toxicology testing and short-term, just like category A, but then we're adding subchronic toxicology studies with rodents, and non-rodents, reproduction studies, developmental studies, metabolism and pharmacokinetic studies. So category C then would be the high toxicological concern. So that would be all of the studies that we just talked about, plus chronic studies, carcinogenicity study, and clinical studies may need to be run also. Okay, so for GRAS, there are some studies that need to be conducted, and either you could do these yourself, or as Bob was saying, they could be out there in the public domain. So genetic toxicology studies, there could be an AIMS, a micronucleus study. There may even be another in vitro mammalian cell assay. So all this really would depend, you'd have to consult with the toxicologist and try to figure out what's going to be needed. Usually we'll just focus on rodent toxicology studies also, and we'll just do 14 to 28 day study. There may be a shorter term study in front of that. It's used as a range finding study. But then usually the penultimate study is a 90-day rodent toxicology study. All right, and then of course we talked about the toxicology studies published in peer-reviewed journals. If you do use those, they do need to be really robust enough to pass muster with the FDA. So you want to make sure that they're in good condition, in good shape. And if you find that there's things that are missing, you might need to go ahead and supplement that and do your own study. All right, so non-clinical studies. So we've got three primary types of studies. We've got pharmacology, which I'm not going to talk about too much. And then we have the pharmacokinetics, and then we have toxicology studies. All right. So now we'll break those down a little bit. Pharmacology, like I said, I'm not going to be talking about this in too much detail right now. There's different types of pharmacology studies. There's the general, just to see if there's an effect of the material, and then there's safety pharmacology studies, just to detect if there are possibly cardiac or pulmonary function, for example. But for the most part, for GRAS, you won't need to do that type of thing. All right, So pharmacokinetics is the next one. And there's 4 items here that we'll go into a little detail on the next slide. There's absorption, there's distribution, and there's metabolism, and then there's excretion studies. All right, so now we'll go into a little bit more detail on these on the next slide. Absorption then would be the process in which an administered material enters the body. So what we could see by that, and what we normally do in our toxicology studies, is we would normally take samples of blood from animals at different time points. And we'd figure out what the absorption in the material was. We'd look at the maximum concentration, we'd look at the time the maximum concentration hit, we'd look at area under the curve, we'd look at a whole bunch of things. And so we would look at that primarily in plasma and serum. And then distribution is where that material or a metabolite goes in the body. You know, does it stay in certain organs, or does it just, you know, do you find it at all anywhere besides the blood? And then after that, we've got metabolism. And that's how the material itself might get structurally changed in the body. Sometimes it'll just pass right through the body, all right? But other times there might be some kind of enzymatic or non-enzymatic reactions. P450s are an example of some enzymatic reactions, but so we're looking at that too. And what we want to do, we do want to make sure that we're using the proper species. And a lot of times with metabolism studies, if you are doing metabolism studies, you can see possibly which animal model, which animal species is similar to humans, produces the same metabolites. All right, and then we'll go on to excretion next. And excretion is the process in which the materials are removed from the body. So you can look at things like urine, feces, expired air. A lot of times it's not done too often with GRAS at all. You don't see that too often. Then pharmacokinetics are next. And well, actually that's still part of it. That's pharmacokinetics are usually done with the absorption phase. And you could, I talked about some of these already, but you could do in vivo or in vitro type studies with pharmacokinetics. So a lot of times we'll primarily do this in animals in the toxicology studies, drawing blood, like I said, and in species. And it is helpful to interpret the results of the tox studies and then extrapolate data from the animals to man, so that you could take a dose that is that there's no effect level in an animal and extrapolate it to man to see approximately where that no effect level could be in man. There's a lot of science and art that goes in there too. So going next to the toxicology studies, and those are studies that are conducted in animals primarily, and they're administered at very high doses to determine toxic effects. And you do want to conduct those according to the GLPs, good laboratory practices. And the reports may be submitted to the FDA. I'd recommend that all studies conducted be submitted in that package, so that the FDA can see that your company has been totally transparent, and you're reporting anything and you're not hiding anything. Okay, so now we're going to talk about some types of studies, some different things you could do in studies. Rodent and non-rodent species may be used. With GRAS, it's primarily the rodent studies. You want to make sure you use the appropriate species, as I mentioned before. And you also want to make sure that there's a sufficient number of animals. Males and females do need to be used for sure. The type and length of study is important to consider. Route of administration for these food products are definitely, they're oral for the most part. And you want to make sure that you know the identity of your material and any impurities or contaminants. So you'd have to make sure that you're going to do analysis, and you'll want to make sure that that analysis is robust, and it'll create an adequate certificate of analysis that the FDA would be happy with. Okay, so if you could click through all of these right now, that first half doesn't work too well for some reason. There are a whole bunch of different types of toxicology studies. The single dose studies, we're going to talk about all these in detail actually, but there's single dose, short-term, subchronic, and you could read this list. A lot of these you will probably, well, some of these you'll do with GRAS, as we talked about earlier, and some of these you won't. So then let's go to the next slide, please. And we'll talk about these in detail now. The single dose studies, you can use those to identify highly toxic materials. If all of a sudden you see that your animals are dying, then you know you do have an issue. So you want to make sure that you might want to run this just to use it as a screen. Single dose is obviously one dose or 1 day's exposure. And then you look at the animals, and in this case, since we're talking about GRAS primarily, you'll look at the rodents, usually rats, and you'll observe them for 14 days. And you'll look at body weights, food consumption, and then there's some other options too. But the body weights and food consumption, you definitely want to look at before and then after dosing, and then throughout the observation phase. And then if we do these in non-rodent species, you probably would want to do a pyramiding or escalating dose study so that you don't have to use a lot of animals. So that type of study, you would start with a lower dose, and then in those same animals, you just escalate the dose with some time in between to clear out the material from the system. And then there's the short-term repeated dose study. So those studies they may detect toxicity in materials that don't produce an immediate toxic effect. So with repeated dosing, you might see something. And for these, you could dose every day, 7 days per week for 1 or 2 weeks. You want to look at observations, just checking the animals to make sure they look okay, see if anything is unusual in the bottom of the cages and that type of thing. You want to look at food consumption and body weights. And then there's some other options you could do too. You could look at, you could do pharmacokinetics, you could do clinical chemistry, hematology, coagulation, water consumption, you could do organ weights, necropsies, microscopic pathology. You could do, and if you do pharmacokinetics, if you are going to be sampling blood from rats and mice, you want to make sure that you have a satellite group, because you can't take too much blood from those animals for serial sampling. Okay, and then the next one would be the subchronic study. And the subchronic study then is that really does detect detect accumulation of materials or other, it could detect accumulation of materials or in tissues or other mechanisms that might be important to identify any toxicity. So you could do this, you dose the animals for 7 days a week for 4 to 13 weeks. You would do, again, observations, food consumption, body weights. You could do electrocardiograms, blood pressure, ophthalmic exams. Usually EKGs are done in non-rodent species. There would be clinical pathologies, pharmacokinetics, organ weights, microscopic, and then other options, which would be on the next slide. So other things that you could do at this point, and this is usually the longest term study for a GRAS, you might want to have a reversal or recovery group that would you would be able to see recovery of any toxic signs. And hopefully, though, you don't have any toxic signs, so a lot of people don't do that part. They're not anticipating any toxicities. You could do special staining, electron microscopy, functional observation batteries, which are required in a food additive petition, water consumption, that type of thing. So after the subchronic studies, we've got chronic studies, and these would be mostly for food additive petitions. They could be up to 12 months in length, dosing 7 days a week. Parameters are really the same for the subchronic, and you could use rodents or non-rodents. And again, similar options as for the subchronic studies. All right, so then if you're doing a food additive petition, you might need to go to a non-contagiosity or carcinogenicity study, same type of study. It determines tumor potential. And this is a lifetime study for rodents. And you would have, the studies could run 18 months to 2 years, and while in rodents, it also requires careful planning and documentation of your experimental design, especially high standard of pathology. You want to make sure that you have a pathologist who's very good at reading slides so they could detect any tumors that are forming, and definitely unbiased statistical testing. You really have to pay attention to those. For food additives, there might be an in utero phase to this too. So you might need to add that too, and your toxicologist could help you with that. So the next after oncogenicity is reproductive studies. And normally we would not do this for GRAS, but for food additives we would. And there's reproductive fertility. We'd be looking at teratology. There's many materials that could affect these parameters. Like thalidomide, for example, they tested it in rodents. They didn't see anything, but once thalidomide was tested in rabbits, they did find that there were issues, as Bob had talked about. So after that, we'll talk about some more reproductive type studies briefly. There's fertility and early embryonic development, and then we have additional ones. We have embryo fetal development, which is also known as teratology studies. It used to be called SEG2 studies. And we have 2-generation studies. So after then, and then there are the other type of tests, food additives. That's probably about all you need. Okay, so after this, then there's genetic toxicology testing, and you would use these studies for GRAS submissions. So these detect materials that exert their adverse effects through interaction with the genes, with genetic material, with DNA. Okay, they could do that directly or indirectly, and they could alter the structure or function. And there's different types of genetic toxicology studies. There's in vitro studies, which is done in cells primarily. Ames is in bacterial cells. Chromosomal aberrations are in mammalian cells, and then mammalian mutagenic assays also. And then there's others. And then in vivo studies could be a mouse or rat micronucleus study. Going back to the Ames, though I did want to talk about that a little more, because the Ames study is one that's definitely done in a lot of these GRAS submissions. They use 2 different types of bacteria, primarily Salmonella typhimurium and then E. coli also. And what happens in this study is the bacteria are mutated to prevent them from making a certain amino acid needed for survival. And then a reverse mutation then would allow the bacteria to grow without that specific amino acid in the media. So if you see the cells growing, if you see a lot of bacterial cells, then you know that you've got some mutation going on. Chromosomal aberrations, they could include, they're directly affecting the chromosomes, and so you could have breaks, you could have inversions, you could have translocations of the genetic material. And then the micronucleus study could be with bone marrow or peripheral blood, and there's different ways that you could do. I'm sorry, that's the mouse. I'm sorry, that was the mouse and rat micronucleus. There are you could do that in the bone marrow or peripheral blood, and they do detect any type of micronuclei that remains after the erythrocyte forms. So going back, there were some other mammalian mutagenicity assays that we're not going to go into right now. And then there are other in vivo ones, which we're not going to talk about now because we don't need to really for this type of, for food application. So then you could also have allergic sensitization studies, and you know, especially if you're doing anything that might have a peptide or protein, you want to make sure that you're identifying any allergic sensitization studies would be with repeat exposure in the animals. Okay, so then you've got all your studies according to your GLPs. Just, you know, out on the internet somewhere where, you know, you don't have to pay to get these expensive studies done, because a lot of them are quite expensive. But you're going to summarize all your studies, and then you're going to take all those summaries and add those to a submission that goes to the agency, and it's got a whole bunch of other things in it too, like, you know, chemistry and history, things that Bob had talked about before. All right, so I'm going to pass this over to Catherine to finish it up.
Great. Oh, sorry. Go ahead, Catherine.
Well, I was going to pass it over to you, Amy, to start with the questions and answers.
Sounds great. I appreciate that. We have a couple that have come in, and the first one is for Bob. And the question is, why is is the Delaney clause still used today?
That's really an interesting, controversial question. The industry and the FDA have been trying to get rid of the Delaney clause since 1959, and because it takes away the authority of the FDA, and of course it costs the industry a fortune when things don't go right. So in '93, I think Clinton signed an executive order which actually embedded risk assessment into the FDA regulations, and it sort of overrode Delaney. But now the courts have been back and forth against this, and they wouldn't let them delete or take Delaney off. There were several seminal papers that were written in the late 90s, and their basic tenet was that the Delaney clause really didn't prevent any cancer. It was mostly macronutrients like fat and carbs and what have you, as opposed to the micronutrients. But oddly, last October, I think the FDA banned some synthetic food additives based on the Delaney Amendment. And it was mostly because they're getting petitions from the activist groups like NRDC and what have you, still stating that the Delaney clause is in effect. So Delaney is in effect, but most of the things are used under the de minimis rule, and the risk assessment is 1 in 1 million. So the FDA still believes that the levels were safe in these things that they banned in October of last year, but because the law still sits on the books, it's very, very much of a political problem.
Okay,
so,
thank you for that.
Yep.
Second question is regarding GRAS submissions made prior to the new regulations. If they haven't been published, are they still GRAS? And is there an expectation for the time of publication?
Go ahead, Bob.
I don't know. Well, if the new regs, if you're talking about the 2016 changes in the seven-step framework, all the things that were done before are still GRAS, yes. And they've just changed the mechanism by the way you put it in. The timing for publications really is journal dependent. It really isn't anything other than that. And how many GRAS of the 847 submissions? Well, it would depend. If those submissions were based on data that was already in the literature, then it was merely a summarization of that in the GRAS application. But if they had to fill in these data gaps with all those studies that Robin was talking about, and spend a lot of money, then they would put them into publication. So some of them have, some of them haven't. I wouldn't know without going through and reading every one of them, which I don't intend to do. So that's my answer.
All right, thanks. Next question is, can you please explain more about three concern levels as a function of cumulative human exposure and chemical structure?
Well, yeah. Well, we had one of the slides that talked about human exposure. So that I guess, you know, I'm not quite sure with cumulative human exposure. I'm not quite sure what the question is there. But with the concern level, if you get to a certain part per billion in the product or of the product for consumption, then that determines what concern level you're at. Chemical structure, you know what? I don't have that information in front of me right now. I'd have to look that up. So I can't answer that right now. But yeah, there is a difference with the chemical structure. They fall into different categories with the three concern levels.
Okay, thank you. Next question is, what is the general time frame that it takes to complete a submission when you already have all the toxicology information?
Oh, that's a good one. It depends on the cooperation of everybody who's working on this. So, you know, it could be if you have people in there that are just, and it depends on what studies you've done. It probably could take just, you know, a few months to get this done, because what you want to do, you want to summarize all of your toxicology studies. You want to coordinate that with the people who are doing the chemistry part and all the other parts. You know, it depends how complicated this is too, because if you have a product that's going into, if you have a GRAS material that's going to go into a whole bunch of different types of products, you really have to look at the consumption that's going to go on in all these different products and try to figure out what the mean intake is, what the 90th percentile is. You know, you want to look at a whole bunch of different things. So it really depends on how complicated your submission is. But I would say minimum. Oh, and then also you want to get this to an expert panel for reviewing. So it depends how cooperative all those folks are too. So I would say minimum of 3 months, probably realistically, but probably closer to 6 or maybe, you know, more.
Probably about right.
Yeah.
There's a lot of work. These are documents that are 100, 150 pages long. So putting all that and getting all the details right and making sure your framework's right and getting everybody to check off the list and get all your expert panel together, it does take some time. But once it's into the FDA, they're turning it around in 90 days. So that's what they're supposed to do. So.
Okay.
Okay, great, thanks. We are running short of time. We've got a number of other questions. So let me just get to a couple here, and then those that we were not able to get to, we'll try to answer those for you in writing. In light of ongoing discussion, what are your thoughts on using only in vitro toxicology tests and human safety data to support safety studies?
Okay. Okay. So, you know, I say this with a lot of people. You know, do you want your children to take this? It has not been tested on any animals, but would you like to try this? Or you're in a human going into a clinical trial, and you have found out that this hasn't been tested in any mammal before. Do you really want to take it? So you know, you really do have to test it in animals, and the FDA is definitely requiring that. You do have to.
So our thoughts might be
unless of course you have prior, there's prior exposure, you know, prior human experience with it. So I don't know if you want to add to that, Bob.
Just going to say what our thoughts are. You know, we should be trying to get away from animals. I know that's the way to go. but this is not what the FDA wants. And if you only go in with in vitro data, they're going to reject you. So you have to do what the rules say, at least for now. That may change because the in vitro testing is becoming more and more accepted for a lot of things, but not so much for GRAS yet.
All right, let's just take one more, and that is there a good resource for a list of approved GRAS ingredients?
Yes, the FDA website.
Yes.
Absolutely, there's a whole list there.
Yeah.
Great. Okay. Catherine, do you have anything to add?
No, I don't. I think that our there are a couple of other questions that we could not get to, and we'll try and follow up with people and get an answer to you on that. We thank everyone for joining us. And if you have further questions, you please feel free to reach out to me at the contact information below. Thank you all for.
Great. Thank you.
Thank you all.